published in the year 2018 and it was published in a journal named Science Advances with an impact factor of 14.14. So the terminology which we are supposed to know is enamel knots, EKs, which are embryonic signaling centers regulating root cusp formation, which is studied in embryology. And PCF is patterning cascade model. It is a developmental framework explaining cusp variations via EK spatio-temporal dynamics. And we have something called enamel dentin junction, EDJ. It is a preserved record of inner enamel epithelium reflecting the enamel knot arrangement. And the Hominoid molars are low rounded cusps which were seen in Paranthropus, which is high cusp gorilla. So it's basically seen in animals. So the novelty of the study is this is the first large scale 3D analysis of EDJ is enamel dentin junction in 763 molars across 70 hominid species. So this tests the PCM. PCM is the patterning cascade model. So this tests the PCM applicability beyond seals or mice to complex primate molars, and they compared it, and the links this links developmental biology, that is the Evo-Devo, to paleoanthropology. It offers mechanistic explanation for fossil dental diversity. And the aim of this study was to assess the PCM explains accessory cusp variation in hominid molars, that is comparing our animals with humans. And the objective was to quantify the enamel non-spatial relationship through the enamel dentin junction morphology and to correlate the intercusp distances with cusp present size. So it also evaluates the PCM universality across hominids and deviations, like with deviations like Caraveli's cusp. So the materials and methodology. The sample comprised of 763 molars with 276 upper and 487 lower molars from 17 different species, which included both animals and humans. And extinct taxa were used like Homo sapiens, which includes recent with the Pleistocene, and Pan species is the gorilla species, basically Pan and gorilla, they come together. Pan troglodytes and Pan panspis, gorilla species, and Pongo species, all basically monkeys. And fossil taxa was also included, which included Australopithecus, Paranthropus, and early Homo sapiens, plus Neanderthal species. The methodological workflow was first the imaging was done using micro-CT. So the 3D surface models, the EDJ was studied. Software, they used Aviso or Amira's software for placing the landmarks. And morphometric analysis was done. Morphometric analysis was based on the dentin horn tips, that is the enamel knot proxies were determined, and the intercusp distance was also analyzed using the Euclidean distance which was used it was normalized to the EDJ surface area. The tooth size correction was studied, and the trait scoring was done using standardized protocols, and the hypocones, the non-metric traits were studied, and ordinal scales where it was segregated accordingly whether it was absent or suspected or present, that is the degree of expression was studied. And they carried on, after the methodology, they carried on the statistical analysis. The key tests which were used were Kenda-Grant correlation test. So that was used to assess the relationship between the cusp presence or the degree of expression aspect and the relative intercusp distance. So significant distance was seen. The p-value was lesser than 0.05. Hypocone, that is upper molar, there was a negative correlation, which indicated that there is smaller spacing. That's a higher likelihood. Lower molar cuspi, it showed a positive correlation with p-value of 0.55 in Homo sapiens. Larger spacing, it means that it is more frequent. And lower molar cusp 6, it also showed a positive correlation similar to lower molar cuspi. So that was also significant, and it was seen across all genera. And non-significant results are also observed pertaining to Caraveli's cusp, except in the Australopithecus species, which was minus 0.31. And for 7 values, it was also, the p-value was significant, but it was not significant. And order logistic regression was studied. So it confirmed the PCM predictions for central cusp, and odd ratio was also studied. So there was a 1.5 to 2% times more likely per unit increase in hypochondrid endochondrid distance. So then subgroup analysis was also done between the fossils versus the extant population. So stronger PCM adherence in Paranthropus and Australopithecus species were observed, that is basically in the animal population. So the molar type, the trends were consistent across all positions, and the results indicated that the hypophone showed a very strong negative correlation with the intercusp spacing. So if the distance is small, it says there is higher vessels. So the distance was small in the graph, and the lower molar cusps 5 and 6, it showed a positive correlation with the spacing. So larger distances means more frequent or larger cusps. The double cusp 6 was observed in the Paranthropus species and Australopithecus species. So it linked to expanded hypochondrial tuberculum sextum spacing. And exceptions to PCM is we had Caravelis cusp, which had no very weak or no correlation with spacing, which was only significant in gorilla species and Australopithecus species. So cusp size was also not significant. And taxonomic patterns were studied. The Molar of Paranthropus or Australopithecus species, it had higher accessory cusp frequencies, which was consistent with the previous PCM frequencies predictions, and higher cusp molars as said, which was seen in gorillas. And Neanderthals, they had large hypophones which aligned with the PCM. Recently, Homo sapiens, it shows reduction due to all processed food and stuff. And the implications for evolutionary biology. So PCM offers a predictive framework for interpreting fossil level morphology, reducing over-reliance on the homoplasy-prone traits in phylogenetics. So it highlights how developmental constraints reshape the evolutionary pathways, like Bunodonty and Paranthropus as an adaptive workaround. And for paleoanthropology, it cautions us against the accessory cusps like Caravelis as taxonomic markers without developmental context, and it reinforces the importance of Evo-Devo approaches to understand the fossil diversity among all species, and it can be used for genetics and development. The deviations, for example, B. Caravelis cusp correlation suggests us a gene environmental interaction deficit like FOX13 gene or nutritional stresses. These can be studied. So for this study, they have used the Straub and tripod framework. So this is observational plus an evolutionary biology study. So the study design was very good. The sample, only 17 species were included, out of which very few Homo species were included. And otherwise the data collection, the protocols were all detailed, and the criteria was well defined. The confounders were not addressed. The tooth size was corrected, but growth rates is assumed a constant only. And statistical methods, non-parametric tests were justified, but multiple testings were not addressed. So for no correction for 7 cusp types were tested, and the results, the raw data is said to be only available on request, so that was a limitation. And otherwise the interpretation was really good. It explained 80% less of the variation, and the mutations, environmental stress, those were all addressed, and the reproducibility pertaining to the landmark placement can be done, but the custom Python code which they used was not provided, since they said it's only available on request. Ethics, the museums and institutions which were used were acknowledged, and the impact is it linked the EK dynamics to the macroevolution, and it definitely had an impact pertaining to this paleontology, genetics and Evo-Devo models. Thank you.